So my question is, how are you dealing with multi-alignment? Because I know novel break actually align contig to the reference. Because the multi-alignment may be quite informative to the copy number variation. I'm wondering how you guys dealing with the multi-alignment in your pipeline.
There's no way to dealing with multiple. We try to get longer contigs, try to resolve the mapping issue as far as we can. But there is still no way, except you get longer sequence. That's my answer.
Yeah. And I'll just touch on that. In point mutations, we remove all reads that actually that would map to more than one location, therefore reducing our false positives. So we don't look at those.
So basically, you just remove all the module. Okay.
Any other questions? I see one. Yes, please.
Okay, I'm not a researcher in this field, so I ask a simple question. So what's the rationale behind the camera? So how camera discriminate between the cancer sample and the normal sample in the sequence, I mean in the structure variance?
As I mentioned, that if there is a breakpoint, it can exist in the, if it exists in the read, there's must some chimeras that contain that breakpoint. A breakpoint is, you can imagine that like a single base resolution. So why we use the chimera strategy? Because chimeras are short and they are error-free, almost error-free, not totally, but they probe to error-free. And we can use a mature chimera hash methods to characterize those chimeras. And the filtering against the reference and the normals could obtain the novel chimeras that we are interested in. So I don't know if I answered.
Okay, another question, a related question. The key, I think, is a different number. So
yeah,
which number is like 3, 4, 5 or more? So which?
Yeah, it's an odd number and it must be smaller than the read length. And usually we in the default is 27, and also 31. We tried a few chimeras to do the analysis. Yeah.
Okay, thank you.
So a question for Paul. Do you, can you tell more about the ways in which you score the structural variants, and a few words about how you will score the actual tumors?
Yeah, sure. So the scoring of the structural variants is actually surprisingly complex. As an example, imagine that the truth was one large event and a group called 3 smaller events within those. We have to decide on how we're going to do that. Do we call all 3 false positives? What if their breakpoints are really close on the 2 ends, but they find something aberrant in the middle? And so I don't think there is an exact right way of doing this, but the way that we did it is we took a look at the base pair span for the predicted structural variants and asked how many of those bases were captured by the truth. So it's kind of like a fractional base overlap. There are several different scoring metrics that we tried, and they don't really change the order of the submissions. So they reflect different aspects of what you prioritize and how you think of things, but they don't change how well different algorithms ran, although they might change error profiles a little bit as you do that analysis. And so that's something that we're digging into now. Your second question was how would we do the scoring of the real tumors? Right, so the way it's happening is that for, let's take single nucleotide variants, we're doing deep resequencing on a large fraction of the predicted variants, and at each one we'll have 300 or 400x coverage, and we'll use a different caller on the alternative technology to predict whether it's there. We'll impose a threshold on the observed allele frequency. So if these are 1 or 2% events in the 300x, then we will exclude those, and then we'll use those to assess a truth and a positive. For the structural variants, we're PCRing around a breakpoint, so we should be able to detect presence or absence of a specific breakpoint, and we'll have pairs of breakpoints that create the events, and we'll see if an event is true because all of the breakpoints that are predicted by the event are actually there. Any other questions? All right, thank you very much.